Hello everyone, this is Nalini DJ of Maharani Science College for Women, working as botany lecturer at Department of Botany. Today I would like to tell you about the nomenclature and classification of enzymes as session 1 of paper 8 Plant Physiology 3. Let us see what is the meaning of enzymes. We all know enzymes are the biocatalysts, the catalysts which are going to help in the acceleration of the biochemical reactions. During acceleration of the biochemical reaction, the enzymes, which are nothing but the protein molecules, which will help the substrate molecules to undergo catalysis to give the products. Coming to the history of enzymes, enzymes are first initially discovered by Louis Pasteur while he was working with the zymase and also yeast cells. He found out the enzyme. First, the word enzyme was coined in 1878 by Frederick Wilhelm Kuhn. The enzymes are nothing but the biocatalysts that are previously been called as ferments. Since they were working with the yeast cells, they were called as ferments initially. This enzyme is a Greek word which tells us en means in and zyme means yeast. That literally means in yeast. So enzymes were first discovered while working with the yeast cells. Coming to the introduction of this topic, enzymes, as we all know, they are very much essential proteins which are going to help in the catalytic activity of the biochemical reactions. These are the organic substances, since they are proteinaceous. And Robert Sumner in 1926 he first purified the enzyme and revealed its protein nature by separating the protein using electrophoretic technique, etc. It is wholly or partially the proteinaceous substance, except the RNA enzymes are ribozymes. These enzymes are also associated with some non-protein part that is called as the cofactor, which may be a coenzyme or the prosthetic group, that will assist the protein part, which is also called apoenzyme part of the enzyme, to work better. And also they are synthesized by the living cells under the direction of DNA in the nucleus. And also sometimes mitochondria and chloroplast DNA also helps in the synthesis of enzymes. They have very much high degree of specificity, that is at most important property of the enzyme, and they function in the dilute solution, aqueous solution, and they are very much specific even with the range of pH and temperature for their working. Too much heat or higher temperature, they are going to get denatured, as do other proteins. Other proteins also undergo denature, so they will get denatured. Let's move on to the next one, that is nomenclature of the enzymes. Nomenclature, as we all know, is the naming system of the enzymes. Enzymes undergo a very important classification groups from the earlier times. Earlier enzymes they used to name the enzyme based on the suffix, by adding the suffix to the substrate name. Suppose maltose is the substrate, then OSE of the maltose is removed and the suffix ASE they used to add. That is the older system of classification. And also at older times they used to name some enzymes based on the source from where the enzyme is obtained. Here, based on the source, we can take the example bromelain is the enzyme which is obtained from pineapple, which belongs to family Bromeliaceae. And also pepine. Pepine is the source of pepine is the papaya. So the name. So based on the source also they used to name the enzymes. This is also an older system of classification. And also the naming system was also followed based on the mode of reaction the enzyme is going to take up during catalysis. Like dehydrogenase. Sometimes they used to add the substrate name also. That is why they are called as succinic dehydrogenase. Here some exceptions are there, that is pepsin, trypsin, pepine are the exceptions to this. Then coming to the other new system of classification that we are following today, that is according to International Union of Biochemistry and Molecular Biology, IUPMB system of classification. All enzymes are categorized into 7 major classes according to this classification. Based on the type of reaction they catalyze. How exactly? The reaction involved, which particular bond has been framed or cleaved. Based on that, the IUPMB classification is a very detailed classification giving all the specific details of the enzymatic activity during a biochemical reaction. So enzyme is described according to this classification with a code word, a prefix, that is EC, enzyme commission number. And this EC number is made out of 4 digits. That is alcohol dehydrogenase is 1.1.1.1. As we can see here, the first digit indicates the major class, classifies the enzyme based on the reaction. The first digit is called major class, that indicates the classifies the enzyme based on the reaction involved in the catalytic activity. And second digit indicates subclass, the type of group which is involved in the reaction, whether it is alcoholic group or any amino group or methyl group, which particular group is involved. Based on that we name the second digit. And third digit indicates subclass, substrate on which group acts. So which substrate is acting on the group, so which substrate has given the cleavage of a particular group in it. Okay, that indicates the third digit of the IUPMB classification. Then fourth digit indicates the systemic specific name of the enzyme and also the serial number of individual enzyme. That fourth digit indicates the serial number of the individual enzyme. There are so many enzymes, no? In that they have given a specific name according to which a serial number has been assigned for each enzymes in IUPMB classification. Let's see how these 4 digits work. In an example, lactate dehydrogenase. Here, first digit indicates which major class it belongs to. It belongs to the class oxidoreductases. Okay, and we can see the second digit that indicates the subclass acting on the primary and secondary alcoholic groups. And third digit indicates the acceptor of that group, the subclass, that is NAD+, which is a cofactor, which is an electron acceptor, cofactor NAD+ as the electron acceptor. And fourth digit, number 27, here it is indicating specific enzyme within the subclass, which particular enzyme. the number, whether it is a dehydrogenase enzyme or sometimes whether it is decarboxylase enzyme, the number will be different. That indicates the fourth digit of the IUPMB classification. So this is considered as the top level classification or most recent classification. So here there are 7 major classes. As we saw here, the class oxidoreductase makes the EC number 1. That is the first class of the IUPMB classification. What are oxidoreductases? Oxidoreductases are the enzymes which help in redox reaction, where the oxidation of one substrate and reduction of the another substrate takes place with the involvement of electrons, removal of electrons. And next one is EC2, that is transferases. Here, as the name indicates, transfer of a particular functional group, whether it is carboxyl group, methyl group, amino group, or phosphate group. Okay, the EC3 is hydrolases, the enzyme which catalyzes the hydrolysis or breaking down of the bonds, like most of the digestive enzyme bonds, glycosidic bonds, peptide bond, ester, ether bonds, using a molecule of water. Those are the hydrolases. Then comes the lyases. Lyases which cleave the bonds by means of other processes except for the hydrolysis and oxidation. They doesn't take place here. They are not going to take place here. And isomerases, EC5, they catalyze the isomerization reactions, that is the shifting of the atoms in the same molecule from one position to another position. Such isomerases reactions takes place. EC6, that is major class 6, is ligases. Ligases is a Latin word which tells us about joining of the molecules. Joining together of two molecules is the ligases. And last and new class is the transferases. So last one is the translocases. Translocases is the newly added one enzyme group to the enzymatic naming or classification. And we can take one more example for the EC number explanation. So here we can take the reduction reaction of the a reaction which is taken up from the glycolysis. That is, here the ATP molecule is going to transfer a molecule of phosphate to the D-glucose. So the product will be the D-glucose 6-phosphate and ADP is released. Since ATP has transferred one phosphate group to D-glucose. The enzyme involved here is exokinase. So exokinase is a transferase enzyme which is going to transfer the amino group. So this molecule, this particular enzyme exokinase belongs to the class transferases. According to the IUPMB system, the name of the this particular exokinase enzyme is ATP glucose phosphotransferase enzyme. This is a complete technical name of this hexokinase enzyme. And this indicates the number, EC number, the 4-digit EC code will be 2.7.7.1.1. Here the first digit 2 indicates it belongs to major class transferases. Second digit indicates it is going to transfer which particular group, it is phosphotransferase. And third number indicates phosphotransferase with hydroxyl group as the acceptor, which group is going to accept the phosphate group in the glucose molecule molecular structural formula of glucose, in which the acceptor will be the hydroxyl group, OH group. And fourth digit indicates D-glucose as the phosphoryl group acceptor. So D-glucose is the substrate which is going to accept the phosphoryl group. So that is how the technical name is going to specify each and every action of the enzymatic reaction. But the trivial name or the commonly used name is the exokinase. And coming to the 7 major classes in detail, we will explain now. So we will see the 7 major classes. So easy to remember. A mnemonic we can have here, that is OTH lilt, or it can also be called as oath lilt. That is the mnemonic that we can follow. Coming to the first major class, that is oxidoreductases. Oxidoreductases are the enzymes which catalyze the transfer of H plus atom or electron pair from one substrate to another substrates. So they are redox enzymes. According to IUPMB, the enzyme belongs to class 1 and designated as EC1. Here EC 1.1, where the alcoholic group is the donor, and NAD plus or NADP plus, those are the coenzymes involved. That will be the third digit, which is going to act as electron acceptors, NAD plus or NADP plus. Let's see an example of oxidoreductase enzyme, that is lactic dehydrogenase. This lactic dehydrogenase and also glucose 6-phosphate dehydrogenase, cytochrome oxidase, alcohol dehydrogenase are the very important oxidoreductases. We can see simultaneously the oxidation of one substrate and reduction of the another substrate takes place with the involvement of 2 electrons, or a pair of electrons will be removed from the substrates. That is the reduction reaction. And coming to the other major groups of oxidoreductases, those are oxidases and also peroxidases, oxygenases, reductases, hydroxylases, and also catalases. These are the some further classification of the oxidoreductases. Coming to the next major class, that is transferases. As the name is suggesting, they are going to transfer a functional group, whether it is a carboxyl group, methyl group, amino group, or phosphate group, except for the hydrogen atom. Hydrogen atom transfer doesn't take place here. So the groups usually transferred by these enzymes are ethyl, methyl, amino, etc. So we can take the example here, AX, that is the group is involved with the substrate A. When it is added with substrate B, then that X group will be transferred to the another substrate, that is B. So A plus BX. This is the schematic representation. And this belongs to EC2. So EC2 enzyme, the enzyme which transfer the 1 carbon group here, and 2.1.1, then the group transfer is accepted by the coenzyme. Which particular coenzyme? Here usually nicotinamide is the coenzyme. Then majorly these enzymes are grouped into 2 major types, that is transaminases and kinases. Aminases which transfer the amino group, kinases which transfer the phosphate group. Other examples being acetyl transferase, methylase, polymerase, exokinases, etc. We can take one more example, that is glutamic acid is going to transfer an amino group to pyruvic acid to make the alanine and alpha ketoglutarate. Here the enzyme involved is glutamate pyruvate transaminase, because glutamate is transferring to pyruvic acid, pyruvate to a group of amine group. So it is called as glutamate pyruvate transaminase. Next major class according to IUPMB is the hydrolases that belongs to EC3. Hydrolases catalyzes the breakdown of bonds or cleavage of bonds of peptide, glycosidic, ester and ether bonds of the substrate, very importantly by utilizing a molecule of water. So A and B are the two substrates which are going to utilize, they have made a bond, and that bond, whether it is a peptide bond, glycosidic bond, ester or ether bond, that will be cleaved or hydrolyzed using a molecule of water. So AOH plus BH will be formed. Since almost all the digestive enzymes, they are made with the dehydration process, that's why they require a molecule of water for enzymatic activity. So bond here in case of starch, in presence of amylase, they are going to make the products, that is maltose, isomaltose and dextrose using the enzyme amylase. Starch is undergoing hydrolysis utilizing a molecule of water. Other digestive enzymes like pepsin, trypsin, urease, proteinase, glycosidase are all the very good examples for hydrolases. Alpha amylase, chymotrypsin, acid phosphatase and alkaline phosphatase are the other examples of hydrolases. So we can take the nomenclature EC3 hydrolase, carboxylic ester hydrolase. So here it is, ester group has been cleaved or broken. And last digit indicates the trivial name, that is glycerol ester hydrolase will be the complete name, but the trivial name is lipase. So that is how we classify, we name in case of IUPMB classification. Then the fourth major class, that is Lil, Oath, Lil, L, first L we have taken, that is lyase enzyme. Here the bonds are going to break without the utilization of water. Here mainly hydrolysis and oxidation doesn't take place. Here a removal of a group from one substrate to another substrate takes place with other mechanism. You can see here the schematic representation. The removal of one group, that is the forward reaction, may involve one enzyme and the backward reaction may involve another enzyme of another major group. And finally it is going to make a product with a double bond. So that those are the lyases. We can see fructose 1,6-phosphate in presence of fructose 1,6-diphosphate. With the involvement of the enzyme aldolase, it is going to give two molecules of 3 carbon compounds, 3-PGAL and also dihydroxyacetone phosphate. Other enzymes which are going to make double bond in the products are aldolases, fumarases, histidase, carboxylase, ketolases, etc. So what are lyases? Lyases are the enzymes that catalyze the addition or removal of a group from the substrate, but without involving hydrolysis or oxidation reaction, and it creates a double bond in the products. We can see here the backward reaction is assisted with the enzyme, that is synthetase, which belongs to other major class, that is EC6. That we can see the involvement of this reversible reaction, two enzymes are involved. Then the next major class, that is class 5, that is isomerases. They catalyze the intermolecular structural rearrangement of the molecule. Isomerase catalyze the transfer of groups within the molecule and they yield different isomeric forms. That is, those enzymes are called as isomerases. These enzymes which catalyze the interconversion of either optical, geometrical or positional isomers by intermolecular rearrangement of the atom or group within the substrate. hence the name isomerases. Major class 5 enzymes are isomerases, epimerases, racemases, etc. Here isomerase is EC5. Excuse me. Isomerase is the EC5. 5.1 will be the epimerase, which particular group involved. And 5.1.1.1, another fourth digit indicates alanine racemase, which particular product is obtained. One more example we can take here, glyceraldehyde 3-phosphate. In presence of triose phosphate isomerase, it's going to give dihydroxyacetone phosphate. It catalyzes the intermolecular shifting of the form. Within the molecule there will be intermolecular shifting of the atoms, or any group, functional group takes place in case of isomerases. Other examples of isomerases being triose phosphate isomerase, phosphoglucomatase. All matases, epimerases, racemases are the good example. Phosphoglucomatase, triphospho isomerase, phosphoexose isomerase are the examples. We can see here, this is the substrate which is going to shift C from last position to middle position. Likewise, the intermolecular shifting of the atoms takes place. Then the last but one, that is the sixth major class, that is ligases or synthetases. It has come from the word ligase, Latin word that is joining together. Till the fifth class, we saw only the cleavage or breakdown of the bonds, but ligase are nothing but synthetases. They are going to join the bonds between the substrate molecules, and they will give a new molecule, synthesize a new molecule. Okay, here the enzyme catalyzes the formation of the bond, such as carbon-carbon, carbon-nitrogen, carbon-sulfur, carbon-oxygen, etc., utilizing the energy which is simultaneously obtained by breakdown of the pyrophosphate bond from ATP molecule or any other compound like FAD, like that any other cofactor can be involved. Example, DNA ligases, glutamate synthetases, pyruvate carboxylases, etc. So here you can see, sorry for that, the arrow has gone little higher. So here A plus B plus ATP will give AB plus ADP plus pH. So A and B, two different substrates, are joined together with the help of the ligases. So ligase is EC number 6, and CO group is joined, then we call it as 6.1. Like that, if other group, they will give another number 6.2, like that. The 6.1.1, that is the third digit, indicates amyloacyl tRNA synthetase, and which particular group is assisting in the joining. Here it is amyloacyl, and it is involving tRNA and synthesizing the compound. That is why aminoacyl tRNA synthetases, like that we name according to IUPMB classification. Next, the last one, that is translocases. It is newly added major class in the IUPMB. These translocases catalyze the movement of the ions or molecules across the membrane. They will help in the movement of the ions within the membrane and their separation within the membranes. For example, enzymes catalyzing the translocation of hydrons H plus and also inorganic anions, amino acids and peptides. Also, carbohydrates and their derivatives are also involved with the translocation activity. And ATP synthase is an enzyme which is going to help in the movement of the molecules across the membrane during the catalytic activity. These enzymes belongs to EC7. This enzymes of reaction that provide a driving force for the translocation linked to reduction reactions. Hydrolysis of nucleoside triphosphate, hydrolysis of diphosphate, and also decarboxylation reactions. So in case of that is tricarboxylic acid cycle or Krebs cycle, the involvement of the translocases can be seen, that is fumarases, etc., the enzymes which are taking the assistance of this last major class, that is translocases. Next, coming to the special group of enzymes and some interesting facts about enzymes. Special groups, the first one being isoenzymes or isozymes. That means the same enzyme in different forms. Enzymes which have similar biological action but they exist in more than one form. They are nothing but different molecular structure of the same enzyme. Hence, isozymes are nothing but different molecular species of the same enzyme. Example, cytochrome, lactic dehydrogenase, etc. Then coming to the zymogens, very important. Zymogens are nothing but the inactive state enzymes. Enzymes, all enzymes, especially the digestive enzymes, will be present in an inactive state in our lumen of the intestine or stomach. Because if the enzyme is in active state without the food in the stomach, then it starts eating up the stomach lining, which is made out of plasma membrane with proteins and lipids. That's why the enzymes all the digestive enzymes will be present in the inactive state. These inactive state enzymes are nothing but zymogens or proenzymes or preenzymes, another name for zymogens. They are proteins with the inactive precursor of the enzyme. They are present as inactive precursors, proteolytic enzymes and also other carbohydrate digesting enzymes. Okay, here the other examples being pepsinogen for the enzyme pepsin, chymotrypsinogen for the enzyme trypsin, that can be seen. So we use the suffix gen, pepsinogen, trypsinogen for the inactive precursor of the enzyme. So such enzymes are called as zymogens. And coming to the other type of enzyme, that is constitutive enzyme. Here the enzyme which is always present in the body in a particular proportion, not going to get synthesized only during emergencies or only when the need is there, but they are present always. Such enzymes are called as the constitutive enzymes. All the enzymes of the glycolysis step, in glycolysis various enzymes comes now, all those are the constitutive enzymes. Always they must be present because it is a very important step in the oxidative respiration. Okay, inductive enzymes. The next one is inductive enzymes. Here, these are the enzymes which get synthesized only when the inducer is present, when the need is there. Inducer is the substrate molecule. Only when that substrate is there, then only the enzyme will be synthesized. So, these are the enzymes which appear in cells when they are needed. Under emergencies, they will get synthesized. Okay, very good example is beta galactosidase of lactoperon. When the lactose is present, then only the beta galactosidase will get synthesized to convert the lactose into D-glucose and also galactose. Then coming to the next interesting enzyme group, that is the rennet tablet or rennet complex. These are the enzymes which are synthesized basically in the stomach lining of the calf, baby cow. Okay, they are secreted as a complex of 3 enzymes. They are renin, protease and chymosin. These enzyme complex is utilized in the commercial cheese making. So this is the initial one. Nowadays even the vegetable rennet complex has also come. Then coming to the one more fact, that is turnover number. We never tell so many milligrams of enzymes are there in the system, but we will see the enzyme proportion based on the rate of reaction, based on their action, how many number of substrates are converted into products. Based on that, we use the word turnover number. For example, here the molecules, number of substrate molecules which acted upon one enzyme in 1 minute. That is the definition for turnover number. Carbonic anhydrase is one such enzyme in which 6 millions of substrates act upon that enzyme in 1 minute. So this is considered as the fastest enzyme. Let us see some other important facts. The fastest enzyme, as we saw, it is carbonic anhydrase, which helps in the carbonic acid formation and also breakdown. And slowest enzyme is the lysozyme, which is present in our saliva and also in our tears. So it is basically a bactericidal enzyme which is very slowest, because the number of substrate act upon this enzyme per minute is very, very less. Then which is the largest enzyme? Largest enzyme is the catalase enzyme, which is present in peroxisomes. Slowest enzyme is peroxidase enzyme, or also called as oxidase enzyme. This is also present in the organelle peroxisomes. So with this, we will wind up this nomenclature and classification session. Briefly, I would like to summarize. These enzymes are such important biocatalysts, which act more than 5000 biochemical reaction types, which act on 5000 biochemical reaction types. These biocatalysts are always proteins. Sometimes only the exception is the RNA enzymes or the ribozymes. They are needed in very, very smaller proportions. And we were using earlier the names, classifying or naming system by adding suffix ase to the substrate name, like maltose enzyme is the maltase, and also the other enzymes, digestive enzymes, ase we used to add. Sucrase, like this, maltase, lactase, like that. And the enzymes which are named later based on the reaction type and also the substrate name, like lactic dehydrogenase or alcohol dehydrogenase, like that. And the most advanced system that we are following for the naming is the IUPMB system of classification. We have seen some special groups of enzymes also, and also about turnover number in this session. And these are few references you can go through. The most familiar question that is asked from this session is classify the enzymes with examples. And also you can go through the MCQs that are very important for competitive